The overarching aim of this procedure is to immunolabel proteins of interest on the outer mitochondrial membrane with an antibody of your choice and analyze concurrently with mitochondrial function. This is accomplished by first collecting the tissue of interest, such as the brain, spinal cord, or liver, from a rat. The second step is to isolate the mitochondria from the tissue, in this case the spinal cord. The spinal cord is homogenized with a Downs homogenizer, and other organelles and cell debris are removed through a series of centrifugation steps until a crude mitochondrial pellet is achieved. Myelin, a major contaminant of central nervous tissue, is removed by mixing mitochondria with a solution of 12% OptiPrep and centrifuging once again. Next, the isolated mitochondria are labeled with a primary antibody of choice, followed by a secondary antibody conjugated to a fluorophore. The final step is to add fluorescent dyes to confirm mitochondrial identity or other functional parameters. Ultimately, flow cytometry is used to show the relative abundance of mitochondrial outer membranes throughout normal physiology or disease.
The main advantage of this technique over existing methods, like microscopy, is that it permits high throughput and rapid quantitation of thousands of individual mitochondria.
Animals used in this study were treated in strict accordance to a protocol approved by the CRCHUM Institutional Committee for the Protection of Animals, which follows national standards as outlined by the Canadian Council on Animal Care. To begin this procedure, place a hole in